And now, without further ado, let's get into how all of this applies to biological systems, why the genetic code is a literal code, and see if this standard atheist talking point can be annihilated once and for all. Now, to do this, rather than just explain this with my own words, I'm going to let the words of experts in the fields of genetics, information theory, computer science, and other relevant fields make the case for me. My objective is simple. First, online atheists attempt to deflect their own ignorance of the subject by accusing me and others of not having the appropriate credentials to be taken seriously, and therefore any argument being made is nothing but a God of the Gaps standard excuse. Second, by the end of this discussion, it is my intent that all who oppose the premise of a literal genetic code being necessary for life to exist must formally debunk the concepts and positions put forth from which my understanding has been developed. For any uninformed or perhaps willfully ignorant evolutionary biologists, biochemists, grad school students, or any other expert in paleontology, primatology, virology, etc., who claim I am ignorant or that the points I make have no substantiation, well, following this, you will have plenty to work with. Make your claim and let the world know which of these concepts you formally reject, which expert by name you didn't know what they were talking about, and most importantly, provide the proof which negates my position. As a foundation to this, let me quote the words of Hubert Yockey from his seminal work, Information Theory, Evolution, and the Origin of Life. Information, transcription, translation, code, redundancy, synonymous, messenger, editing, and proofreading are all appropriate terms in biology. They take their meaning from information theory and are not synonyms, metaphors, or analogies. Now, we will go deeper into one of his papers here in a few more minutes, but to lead off, I want to substantiate my position that information and communication theory has direct importance to biology, in spite of the fact that it seems many evolutionary biologists operate from the perspective it has no bearing on their field of study. So now reading from Should Genetics Get an Information Theoretic Education by Gerald Batal. The many successful applications of information theory now provide strong experimental proof of its validity in the field of communications. Although it became the most powerful conceptual tool available in this field, it remains almost unknown outside the communication engineers community. It shaped much of the present way of living, but few people realize this even though everybody makes daily use of its engineering products. That information theory is largely unknown in the biologist community does not mean that the importance of information in the living world is overlooked. An increasing number of biological papers are devoted to many forms of information recording and transfer, and the word information has become ubiquitous in the biological literature. However, it is used most often with a loose meaning. Many biologists seem to ignore that the scientific concept of information generated a science having reached maturity. We believe that no real progress will result from the recognition of the prominent role of information in life phenomenon, unless information theory is integrated into biology, as physics and chemistry have been. Now, it's rather obvious to anyone paying attention that Batal is saying, hey everyone, it is now proven beyond doubt that information theory has direct implications for genetics, even to the same extent as physics and chemistry. However, it seems like many biologists aren't really grasping this or are refusing to do so. Does that sound like any Evo atheist activists here on YouTube? Up next, I'm going to just show you a chart directly from the paper A Binary Representation of the Genetic Code by Louis Nemser. I won't read from it, but I want you to see for yourself that binary code is directly applicable to the genetic code. And this actually lends itself to something I won't discuss in this presentation, but we now have modified existing code such as the Verilog machine code and done a code mapping to genetic code. This and several other coding languages are currently being used to generate genetic programming in biotechnology. It's actually become quite a big deal and is one of the main reasons there is so much recruitment of programmers to go work in biotech. Now, let's continue on, and it's time to get into some more of the details of the genetic code, and I want to underpin something I referenced earlier in relation to the Shannon-Weaver model, and that is the three recognized components of information itself and their rules: syntax, semantics, and pragmatics. These factors in relation to the genetic code are well explained in a 2017 paper titled Two Genetic Codes: Repetitive Syntax for Non-Binding, Non-Coding RNAs, Non-Repetitive Syntax for the DNA Archives by Gunther Witzani. The code without commas consisted of a nucleotide alphabet with 4 characters and their complementary bases storing biological information to be translated into proteins by living cells. Later on, Manfred Egan insisted that this genetic code represents a real language and not just a metaphor. At this time, it was thought that every natural language or code was determined by the rules of its syntax, i.e., how the single characters could be correctly combined into a lineup to build extended sequences of information. The meaning, semantics, of a certain DNA sequence is defined by the protein product as the result of gene expression, transcription, and translation into the amino acid code. The concept of Noam Chomsky of a context-free universal grammar being present in the nucleotide sequences inspired bioinformatics to study what Manfred proposed in the early 1970s. Life is physics and chemistry and information. But in the 80s of the last century, empirical evidence proved the fact that natural languages and codes are determined by a third level of rules besides grammar and semantics, namely pragmatics. Pragmatics represents the level of rules that interconnect the real character users of a natural language or code with its real-life situations. This means the context in which a natural language or code user is interwoven. This became clear with the empirical fact that no natural language speaks itself as no natural code codes itself. In any known case, there are living agents being competent to generate and use a natural language or code. Pragmatics, therefore, represents the context dependence of meaning, which was formerly presumed to depend on the syntax structure of a natural language or code sequence. Epigenetics proved this fact, that it was not the nucleotide syntax alone which defines the final protein product, but the epigenetic markings. Hopefully that was all fairly straightforward and easy to understand, but to reiterate, it is now recognized that syntax, semantics, and pragmatics all exist in the genetic code. It is not possible to define all of these things inside of a code unless the code actually exists. Anyone who says otherwise is either ignorant or desperate to avoid acceptance of reality. However, I want to expand upon this and remove all opportunity for semantic games. One of the points I make on a regular basis is the arbitrary value assignment for alphabets. In case you haven't been paying attention, this exact point is part of the entire code challenge. Moreover, and I cannot stress this enough, the ad nauseum talking point about deterministic physics and chemistry explaining all of this has been annihilated. Continued usage of it just showcases willful ignorance or just outright stupidity. So, to remove all doubt upon this, let us now read from a foundational paper by Marcello Barberi. What is code biology? We must face the problem of the reality of the organic codes. Are they real codes? Do they exist in living systems? It is a fact that the genetic code has been universally accepted into modern biology. But let us not be naive about this. What has been accepted is the name of the genetic code, not its ontological reality. More precisely, the genetic code has been accepted under the assumption that its rules were determined by chemistry and do not have the arbitrariness that is necessary in any real code. More precisely, the genetic code has been accepted under the assumption that its rules were determined by chemistry and do not have the arbitrariness that is essential in any real code. The theoretical premise of this assumption is the belief that there cannot be arbitrary rules in nature, and this inevitably implies that the genetic code is a metaphorical entity, not a real code. This idea has a long history, and let us not forget that for many decades it has been the dominant view in molecular biology. The very first model of the genetic code was the stereochemical theory, an idea proposed by George Garneau in 1954, and later reproposed by many other authors, which states that the relationships between codons and amino acids were the result of stereochemical affinities. The second canonical model was the coevolution theory proposed by Wang, according to which the genetic code co-evolved with the biochemical pathways that introduced new amino acids into protein synthesis. Both theories imply that the rules of the genetic code are chemically determined and no arbitrariness exists in them. It has taken a long time and much experimental work to overturn this conclusion, but eventually it has been shown that there is no deterministic link between codons and amino acids because any codon can be associated with any amino acid. This means that the rules of the genetic code do not descend from chemical necessity, and in this sense, they are arbitrary. Today, in other words, we have the experimental evidence that the genetic code is a real code, a code that is compatible with the laws of physics and chemistry, but is not dictated by them. Our problem, therefore, is to take stock of this reality and to account for it. I could stop here, but as clearly stated, my objective is to leave no doubt regarding this topic. So let us now expand upon the concept of code mapping, which was discussed earlier, and the necessity for adapters in relation to translation. So now continuing on from the same paper. A code has been defined as a mapping between the objects of two independent worlds that is implemented by the objects of a third world called adapters. The genetic code, for example, is a mapping between codons and amino acids that is implemented by transfer RNAs. A single transduction code is a mapping between the first and second messengers that is implemented by molecular receptors. The Morse code is a mapping between the letters of the alphabet and groups of dots and dashes that are implemented by neural circuits according to a set of learned rules. The key point is that the engineering codes transform a linear sequence, ABC, into another linear sequence, AAA, BBB, CCC, that belongs to the same alphabet world, whereas the genetic code, for example, transforms the linear sequence of nucleotides into a linear sequence of amino acids that belongs to a different alphabet world. The result is that the genetic code and other organic codes require adapters to create bridges between worlds, whereas no such adapters are required in the engineering codes. Okay, so what does this mean, and why do they make the distinction between codes like Morse, the genetic code, and engineering codes, and why does one type need an adapter and the other one doesn't? They answer these questions in the last two sentences, which further supports my argument, so let's read them again. The key point is that the engineering codes transform a linear sequence, ABC, into another linear sequence, AAA, BBB, CCC, that belongs to the same alphabet world, whereas the genetic code, for example, transforms a linear sequence of nucleotides into a linear sequence of amino acids that belong to a different alphabet world. The result is that the genetic code and other organic codes require adapters to create bridges between two worlds, whereas no such adapters are required in the engineering codes. And now, just to reconfirm the arbitrary component of codes, what is essential in any code is the arbitrariness of its rules. And we may well acknowledge that there are arbitrary rules of correspondence between objects that belong to the same world as well as between objects that belong to two different worlds. All right, moving on. Let's get a bit more detailed in relation to the structure of the genetic code. Shannon information, communication systems and messaging, block coding, and its relevance to the genetic code, alphabet extensions, and also why I often use ASCII or Unicode as an example of how all this is applicable. So let's now dive into the 1992 Origin of Life on Earth and Shannon Theory of Communication by Hubert P. Yockey, and just make sure to pay close attention to the non-material aspect of the message versus the measurable information at the very beginning. Like all messages, the life message is non-material but has a measurable information content. Of course, the genetic message, although non-material, must be recorded in matter or energy. A binary alphabet consisting of 0 and 1, each containing one bit of information, is used in the source alphabet of computers and in all communication technology. The receiving alphabets in all applications have more than 2 letters. The binary source alphabet must be extended by forming pairs, triplets, quadruplets, and so forth to form receiving alphabets larger than 2. These alphabets are called the first, second, third, and so forth extensions of the primary binary alphabet. These extensions are called a byte. The use of an initiator codon and codons to terminate the sequence, and the realization that the genetic code is a block code like the barcodes discussed above, makes it unnecessary to assume that commas are needed. The source alphabet of the genetic code is the 4 nucleotides of DNA and mRNA. Thus, each nucleotide has a 2-bit byte. The first extension of that quaternary alphabet has 16 letters. That is not sufficient to code the canonical 20 amino acids that are transcribed into protein. Accordingly, nature has gone to the second extension 64-letter alphabet. Thus, the genetic code has a 6-bit byte, usually called a codon. Sometimes a codon is called a word, but this is clearly inappropriate. The codon is a letter in the second extension of the mRNA alphabet. The protein is a word. The genetic code, shown in Table 2, shares a number of properties with the postal ZIP+4 code and the ASCII computer codes. The genetic code is a block code because all codons are triplets. The genetic code is distinct and uniquely decodable because the single methionine codon AUG and sometimes the leucine codons UUG and CUG serve as a starting signal for the protein sequence and perform the same function as the long frame bar at the beginning of the postal message in zip+4 code. It is a curious fact, highly relevant to the origin of life, that the genetic code is constructed to confront and solve the problems of communication and recording by the same principles found both in genetic information systems and in modern computer and communication codes. The origin of a rather accurate genetic code is a pons asinorum that must be crossed to pass over the abyss that separates crystallography, high polymer chemistry, and physics from biology. As I mentioned above, there is nothing in the physiochemical world that remotely resembles reactions being determined by sequence and codes between sequences. The existence of a genome and the genetic code divides living organisms from non-living matter. At this point, I would hope the clear distinction between living and non-living matter has been established and that no rational individual will continue to spew the assertion that the genetic code isn't really a code in the literal sense. However, I'm sure some will. So let's proceed on to another discovery discussed in a 2017 paper which expands upon the application of contextual interpretation. So the case for the genetic code as a triplet of codes. The efficiency of codon translation in vivo is controlled by many factors, including codon context. At a site early in the Salmonella flagellum gene, the effects of translation on replacing codons THR6 and PRO8 of flagellum with synonymous alternates produced a 600-fold range in flagellum activity. Synonymous changes in THR6 and LEU9 resulted in a 2-fold range in flagellum activity. The level of flagellum activity produced by any codon arrangement was directly proportional to the degree of in vivo ribosome stalling at synonymous codons. Synonymous codon suppressors that corrected the effect of translation defective synonymous flagellum allele were restricted to 2 codons flanking the translation defect codon. The various codon arrangements had no apparent effects on flagellum mRNA stability or predicted mRNA secondary structures. Our data suggest that efficient mRNA translation is determined by a triplet of triplet genetic code. That is, the efficiency of translating a particular codon is influenced by the nature of the immediate adjacent flanking codons. A model explaining these codon context effects by suggesting that codon recognition by elongation factor-bound aminoacyl tRNA is initiated by hydrogen bond interactions between the first 2 nucleotides of the codon and anticodon, and then is stabilized by base stacking energy over 3 successive codons. The genetic code for life is a triplet base code. It is known that adjacent codons can influence translation of a given code and that codon pair biases occur throughout nature. We showed that mRNA translation at a given codon can be affected by the 2 previous codons. Data presented here support a model in which the evolutionary selection pressure on a single codon is over 5 successive codons, including synonymous codons. This work provides a foundation for the interpretation of how single DNA base changes might affect translation over multiple codons and should be considered in the characterization of the effects of DNA base changes on human disease. Every time I read through that, it blows my mind to consider the reality that there is a contextual-based interpretation of a message that is being transmitted through a specific sequence of molecules. It seems that just like what we do in our block codes, as touched upon by Hubert Yockey in his paper, context can both influence and in some cases alter the semantics of a message. And what's even more astounding is that people still argue it's more likely that this happened by chance rather than intelligent agency. However, in spite of everything covered so far, on the off chance that there are still people who want to cling to the uninformed atheist talking point that it's not actually code, let's go one additional layer of what is known and is honestly rather mind-blowing, and that is multiplexed codes. Information processing in genetics is an emerging field. An important field of investigation is the estimation of entropy of DNA sequences. The entropy of sequences was essentially used in order to recognize and characterize various regions of DNA sequences. In these studies, DNA is considered as digital data using a quaternary alphabet. This kind of representation implies also some works at the frontier of digital communications. Lots of studies deal with coding and especially error coding schemes. Nevertheless, another key component in communication systems is the source coding. In this paper, we will compare the genetic code with a recent source coding method called multiplexed codes. Now, quick interjection before we dive into the meat of this paper. You notice that they mentioned error coding schemes and how many studies are being conducted on finding the error correction codes. This is something I haven't even touched on in this presentation, but let's just say it is well recognized that without them, it is not remotely plausible for life to exist. Beyond that, we know error correction is executed, but we are just now scratching the surface of what those codes actually are. Now back to the paper. This paper is organized as follows. First, we shall introduce the genetic code. This presentation will focus on some important features of this code: degeneracy, universality. In section 3, we will present a recent source coding method called multiplex codes. We will note some similarities in the construction of multiplex codes with the genetic code. In section 4, we will go further into the comparison between these codes by considering the genetic data of several species study. The multiplex code described in the previous section has numerous similarities for the genetic code. Indeed, code words in genetic code or codons have a fixed length, as in multiplex codes. Moreover, the number of codons, 64, is much greater than the number of source elements, 20 amino acids and the stop signal. Therefore, these source elements can be considered as the high priority source and are associated to subsets of codons called equivalence classes. This feature was previously called the degeneracy of the genetic code. In our introduction of multiplex codes, code words were composed of binary elements. In genetics, the alphabet used is quaternary, A, G, T, C. This difference is not very important from the conceptual point of view, but appears inversely interesting. Indeed, multiplex codes use the inherent redundancy of a fixed-length code to convey a secondary source of information. We can see that first maxima is reached for a quaternary alphabet, such as the one used in the genetic code. According to multiplex code theory, this redundancy can be used to convey information from a secondary source. This remark can be a new argument in favor of what is the optimal size for the genetic alphabet. All the above remarks show that there is a link between multiplex codes and the genetic code. In order to validate this link, we shall now study the equivalence classes in the genetic code. Okay, another quick explanation of the importance of what they are discussing here. Quickly, just real fast, run through your memory bank and think about the number of times you have heard Evo Atheists talk about how all of these synonymous codons are clearly evidence against the code being intelligently designed. After all, if there was a designer, it would be simple, right? Why all of these duplicate codons? Well, basically what these guys are saying in their paper actually supports the point I make on a very regular basis, which is, well, first off, until recently, we didn't even know what to look for. And as I stated at the beginning of this paper, as they stated at the beginning of this paper, multiplex coding is a relatively new concept. So the premise of applying it to genetics in terms of understanding didn't exist because humans didn't even know about it yet. We hadn't even thought of it. But hey, turns out it actually is directly applicable. So, man, this kind of secondary piece. If you are one of those people still regurgitating the whole standard talking point of, oh, duplicates, therefore no designer, all you're really doing is showcasing how uninformed you are. Now let's get back to the paper. In this paper, we have presented similarities between the genetic code of living organisms and multiplex codes recently developed for source coding of two sources. Multiplex codes exploit the inherent redundancy of a fixed-length code to convey information from a secondary source. In this way, multiplex codes can reach the compression rate of a variable-length codes for the high-priority source while preventing the problem of desynchronization due to errors in the code words. We have compared the genetic code according to multiplex code theory. In our comparison, we have also taken into account some features of the genetic code, in particular the fact that the genetic code is the same for all species. Our experiments show that the genetic code is almost optimal according to multiplex codes theory. Indeed, our study involves the comparison of compression rates. By construction, multiplex codes aim to minimize the mean description length of the source with high priority. We have pointed out that the genetic code is close to results obtained by multiplex codes concerning the representation of amino acid sequences. Moreover, the redundancy of the representation of the high-priority source with a fixed-length code is used in multiplex codes to convey the information from a second source. We have also shown that the redundancy found within the genetic code is close to the one found with multiplex codes. It appears also that the use of a quaternary alphabet for the genetic code maximizes the amount of secondary information conveyed by code words, which is interesting when considering multiplexed codes theory. Wow, let me read that sentence one more time. It also appears that the use of a quaternary alphabet for the genetic code maximizes the amount of secondary information conveyed by code words, which is interesting when considering multiplexed codes theory. So it turns out that not only is the genetic code 1 in a million just as a standard code, it is also able to maximize the transfer of a secondary layer of information. Ladies and gentlemen, I ask you bluntly, how can something be analyzed and found to be optimal when compared to other possible codes, not just for one layer of code, but for a secondary, and not be a code? I mean, honestly, it's getting ridiculous to hear Evo Atheist and even some supposedly educated individuals claim that this is not reality, and then it goes even further when they argue that, oh, if it was, why isn't it binary? Binary's better. No, it's not, ladies and gentlemen. We just weren't able to actually utilize binary when we originally developed our computer technology. But let's go on, and now I'm going to read from Survey of Engineering Models for Systems Biology. This is a very fascinating paper, and it's going to get interesting. Systems biology takes a systems engineering approach to characterize biological processes. As such, systems biology necessarily relies on using modeling formalisms to synthesize data into a consistent mathematical framework. In this paper, we show that the cell is an embedded computing system, and, as such, several modeling formalisms are appropriate for use in systems biology context. Furthermore, this reveals that the engineering disciplines of computer systems engineering, electrical engineering, and chemical engineering are relevant for development of systems biology models. The cell is an embedded computer system because it has the properties of an embedded computer system architecture. It operates concurrently, with various chemical algorithms and autonomous actors acting in parallel. It is reactive and live because an overly slow response will abort chemical algorithms, possibly resulting in the death of the cell. It is robust because it is capable of adapting to changing conditions. Lastly, the system is heterogeneous in that each protein constitutes a unique type of processing element. We acknowledge that a cell is likely more than an embedded system, but it is certainly not less than an embedded system. Okay, the words you just heard obviously substantiate the points I've made throughout this presentation. For how can something that is an embedded computer system operate without source code, without logic rules, without the freely recognized components necessary for information processing? Now, this reality leads you to a decision fork. Based on the evidence presented, you must either come to the conclusion that it is plausible not only for source code to create itself, but also the hardware. Both of these occurred without any direction from an intelligent agent, or they did not. If you conclude the latter, then you must now ask which intelligent agent is the most plausible to have been responsible. If you conclude the former, you must now think critically and ask yourself a simple question. Is it logical, plausible, and probable that these things come into existence by chance, or is it more rational to conclude that an intelligent agent is necessary for them to exist? If so, then the standard dismissals of you don't know how it was done, therefore it must be by chance, or you don't know, therefore God, become so obviously juvenile and petty that these positions, they become laughable stages of immature ignorance that is on full display. Now, the last thing I'm going to get into here, and to be honest, is just to completely obliterate the joke that evolutionary and genetic algorithms, genetic programming, and evolutionary strategy have become when used as supposed evidence for the case of evolution. So let us quickly read from a 2018 review paper on this exact subject. Our analysis of relevant literature shows that no one has succeeded in evolving non-trivial software from scratch. In other words, the Darwinian algorithm works in theory, but does not work in practice when applied in the domain of software production. The reason we do not evolve software is that the space of working programs is very large and discrete. Although hill-climbing heuristic-based evolutionary computations are excellent at solving many optimization problems, they fail in the domain of non-continuous fitness. This is also the reason we do not evolve complex artificial life or novel engineering designs. Now, they're going to continue on, and throughout this entire paper, they use massive citations to support their positions, and here's just one of them. Genetic algorithms do not scale well with complexity. That is, where the number of elements which are exposed to mutation is large, there is often an exponential increase in search space size. This makes it extremely difficult to use the technique on problems such as designing an engine, a house, or a plane. In order to make such problems tractable to evolutionary search, they must be broken down into the simplest representation possible. Hence, we typically see evolutionary algorithms encoding designs for fan blades instead of engines, building shapes instead of detailed construction plans, and airfoils instead of whole aircraft designs. The second problem of complexity is the issue of how to protect parts that have evolved to represent good solutions from further destructive mutation, particularly when their fitness assessment requires them to combine well with other parts. Wow. Well, I would say that is a rather direct and unavoidable critique of the entire premise of what we call life arising through undirected process, new body plan formation through microevolution, and so on, but I shall save the expansion on that for another day.